It's now my great pleasure to introduce the final plenary speaker of the day, Richard Buggs, who is an evolutionary biologist, although aren't all biologists evolutionary biologists ultimately, but Richard is more so than most, and writes on a range of evolutionary issues. But here, he's here as the senior research leader for the Royal Botanic Gardens at Kew, and he's a professor of evolutionary genomics at Queen Mary. He looks at DNA sequences to understand plant evolution, as well as accelerated evolution, which obviously with the climate change pressures that are on us and so on is really important, particularly in the context of this meeting around pests and pathogens. The latest project is on the genomics of ash, looking at ash dieback and the emerald ash borer. I noticed that he has a species of birch named after him, which when I checked it out, I was very jealous to see that it was one of his PhD students who gave him the accolade. So he must also, as being an excellent researcher, also be an excellent PhD supervisor. So without further ado, Richard, it's a great pleasure to hand over to you for your plenary on genomics for healthy trees.
Well, thanks very much, Jerry, and sorry we can't be with you in Birmingham today. But I guess online is next best. And thanks for the invitation to the organisers to speak about genomics for healthy trees. So I guess we're all quite familiar with the idea of genomics being an important part of the future of human medicine. And we're in the early phases of personalised medicine, where people can have predictions of how susceptible they might be to different cancers and how they might react to different drugs, etc. So we're used to the idea of genomics in medicine. We're also very used to the idea of genomics in crop breeding. And of course, a lot of money has been spent on understanding the genomes of the different crops we work with and how that might be used to increase yields and also to increase disease resistance. And of course, a lot of this is very cutting edge research that hasn't yet had as many applications as it will in the future. But less work has been done on the genomics of trees and in the context of tree health. And so that's what we're trying to do in my lab. And we're gaining inspiration both from the way genomics is being applied in medicine and the way in which genomics is being applied in crop breeding. And we kind of look to both, because although you might think, well, a tree is a plant and therefore the situation is most similar to the situation for something like wheat, well, actually, in many ways, the situation is more similar to the situation in humans in terms of the kind of longevity that trees have and the difficulty of doing any experiments on them. You know, if you're working on wheat, it's quite easy to think, oh, here's a gene that I'm interested in. Here's an inbred line. I'll do a cross, and then I'll look at the family and look at the genetics of the progeny. And that, of course, can't be done with humans, and it's difficult to do with trees. They don't have an annual life cycle like wheat. And so in many ways, we have to look to what the people are doing on humans to get inspiration for methods to apply to trees. And so that's been quite fun to have to think through all of these issues. And we live, of course, in a time when globalization has meant that there's massive trade going on around the world. Here's a map that I think I first saw in a talk by Nicola Spence, and I pinched it. Here's a whole load of shipping routes around the world on shipmap.org. And of course, as we ship containers full of goods around, we're also shipping around pests and pathogens. You all know that far better than I do, I'm sure. Well, we're also at a time when we want more trees, partly for biodiversity and partly for carbon fixation. And so lots of governments in the world have objectives to plant more trees. In the UK, we want to treble woodland creation rates in England and increase planting rates to 30,000 hectares per year by the end of this Parliament, a massive objective there. And of course, if we're planting trees and they're just dying from pests and pathogens, then we're not getting anywhere. And so the government has recently launched a Centre for Forest Protection to enhance the protection and resilience of our trees. And that's where my most recent funding has come from. And so the Centre for Forest Protection was launched just a few weeks ago. It's a collaboration between Forest Research and Kew, funded by the Department for Environment, Food and Rural Affairs. And I'll mention some of the new projects that we're just launching at the moment as this talk goes on. And the URL is there if you're interested in finding out more about the new Centre for Forest Protection. Well, when it comes to UK trees, of course, our most commonly planted trees are Sitka spruce and Scots pine. And it would be nice to work on them from a genomic perspective, and people do, but they're quite hard to work on because they have huge genomes. So you can see here that the hectareage being grown of them in the UK, and also their genome size. And basically all of the conifers have massive genomes, 20 gigabase pairs. Compare that to humans, which have just over 3 gigabase pairs in their genomes. Very expensive to work on genomes of that size. Whereas broadleaf trees are really nice in that they have small genomes, much smaller than the genomes of humans. And so wanting to sort of go for the easiest thing, in my lab, we've gone for oak, birch and ash rather than the conifers, so that it's slightly less effort to do genomics research. You can sequence a whole genome of an oak tree for a couple of £100, whereas for a Sitka spruce, it would be several thousand to get the same kind of quality. So I'll talk you through the work that we're doing on those three species, ash, oak and birch. Ash is the one that we've been working on since 2012, when ash dieback was found in the UK, and David Cameron assembled the COBRA committee to come up with a strategy. I'm sure all of you know that it's caused by the fungus Hymenoscyphus fraxineus, and it causes these cankers and causes leaf loss and the characteristic dieback of ash trees. And right at the early stages of the pandemic in the UK, Forest Research set up some mass screening trials. They took saplings that were stuck in nurseries, they couldn't be moved because of the movement ban on ash, and they planted them out at 14 different sites. There were 15 different provenances represented. I've circled in yellow the different native seed zones that the saplings came from. And all of the sites where they planted them were in the southeast of England, where they could be expected to get hammered early by ash dieback, which of course they were. And we went along later and found the best sites, the sites where the trees had established best and ash dieback had done its worst. And we worked with Forest Research to phenotype the trees, scoring them for how much dieback they were showing. And so in 2016, you can see here results for North Birmingham in Norfolk. You can see along the horizontal axis the different provenances that were being grown, one from France, one from Germany, and then others from England, Wales, Scotland and Ireland. And then here you can see the percentage of the trees. And then our scores go from 0 to 7, where 7 is a healthy tree, 0 is a missing tree, and 1 and 2 are different ways of being dead. And there's a continuity of different health categories of living trees there. And you can see that from all of the provenances, there are a small number of trees that were still healthy in 2016, but a lot that were dead or highly damaged. But together with studies in Europe, this suggested that there were a minority of ash trees that had some degree of resistance to ash dieback. So that was to some degree encouraging to see. So what we then decided to do with funding from Defra was we went through those trials and we selected about 625 healthy trees. And we paired each of those healthy trees with an unhealthy tree from the same provenance. And then we sequenced the genomes and compared the healthy and the unhealthy trees. And we looked for alleles, that's variants of genes. We looked for variants where one type was present more often in the healthy trees than in the unhealthy trees. That's a genome-wide association study. And that gave us this, what's called a Manhattan plot, where along the horizontal axis is laid out the ASH genome. It's 880 megabase pairs long. And each of these dots is a single point in the genome, a single letter of DNA that we genotyped. And then the height that they are vertically is how significantly associated variants within them are with healthy trees under ash dieback inoculation. So for example, here, there are a couple of letters of DNA that vary between different trees, and one of the variants is quite significantly associated with the more healthy trees. So this very similar to what's done in human studies, this allowed us to identify something of the genetic basis of resistance to ash dieback. And when we started to look at what was encoded by the ash genome close to these sites that we found associated with level of damage to ash dieback, some of them were homologous, were similar to genes in other plants like Arabidopsis or potato that had previously been identified as genes that had functions that could be to do with pathogen resistance. So this was very encouraging. It made us think, actually, we really could be onto something here. So what we then wanted to do is take that knowledge and be able to predict how healthy or unhealthy individual trees would be under ash dieback. So we trained a genomic prediction model, and now we're being more inspired by plant breeders and animal breeders than by medics. We trained a genomic prediction model on those planted trials, and then we applied that to a set of trees where we had 75 healthy trees and 75 unhealthy trees. And for each of those trees, we predicted how healthy or unhealthy it would be under ash dieback, given its genotype. And we found that overall, the breeding values that we estimated, a breeding value is our estimate of the sort of genetic worth of that tree for a breeding program for resistance. We found that the trees that really were healthy, when we scored them for damage, had on average higher genomically estimated breeding values than trees that were damaged, that were susceptible to ash dieback. And so again, this suggested to us that we really were onto something. Now you can see there's a lot of overlap in these distributions. So for an individual tree, we can't tell you for sure whether it's going to be healthy or unhealthy. But on average, applied across a large number of trees, we can start to separate out trees that really are genetically likely to be more healthy from trees that are more likely to be unhealthy. And this was published in 2019 in Nature Ecology and Evolution, in collaboration with several PhD students in my lab, Steve Lee from Forest Research, and Richard Nichols, who's a statistician, Professor Richard Nichols at Queen Mary University of London. Now, having that knowledge, we knew that that could inform a breeding program, but we also thought, well, if there really is this genetic basis for resistance to ash dieback already out there in ash populations, and given that ash populations are already being devastated by ash dieback, won't natural selection be at work in those natural populations to already be breeding resistance to ash dieback for us? And if it is it working so fast that actually we don't need a breeding program because nature is doing the job for us? And of course, that's quite an interesting question for Defra, because they're thinking about should they fund a breeding program or not. And if there's potential to save taxpayers' money, that is no bad thing. So we looked for a wood where there was lots of ash and lots of natural regeneration of ash and little human intervention, and something not too far from Kew so that we didn't have to travel too far. And we found a really nice site, Marden Park Wood in Surrey, which is owned and managed by the Woodland Trust. Semi-ancient natural woodland, no felling except for ash near paths that have got really dangerous. So we chose a site that's far from any of those paths, with lots of natural regeneration going on. And here's a map of the area of woodland that we studied. And smaller trees are shown by just small dots and larger ones by larger circles. So we surveyed this and we labeled the trees and we collected tissue from just over 600 trees. We scored the juveniles using a similar system to the one we used on the mass screening trials that Forest Research have. And the adult trees we scored by just eyeballing their leaf canopies and estimating the percentage loss of their leaf canopies. A much less good way of estimating, but the best we could do with what we had available. And here I'm just showing a comparison of surveys of the visual symptoms on the trees in the years 2019 and 2021. So here's the adults by percentage canopy cover. And you can see the number of trees with over 75% canopy cover has gone down between 2019 and 2021. So, and that's true of all the metrics. And similarly with the juveniles, health score of 5 is the most healthy trees. The number of the most healthy trees is a lot lower in 2021 than 2019. So this is really showing us two things. One thing is that, yes, the damage is getting worse, the disease is progressing. But we're also seeing that damage for younger trees, for the juvenile trees, is happening faster than damage of adult trees. And that's something we'd expected because lots of studies across Europe have shown that adult trees take a much longer time to die from ash dieback than juvenile trees. And there's probably a couple of things causing that. One is that the adult trees are simply bigger, so the fungus has to grow a lot more within the tree before it can girdle the whole stem of the tree and kill it. And the other thing is that the fungus is producing its spores within the leaf litter on the forest floor after the leaves fall from the ash trees. And so a juvenile tree simply has its leaf canopy much closer to where the spores are being produced, and so get much more infected much more quickly. So given that adult trees are living longer than juvenile trees, we thought, well, hopefully the adult trees represent to us the population that existed before ash dieback came into the country in 2012. It gives us a good idea of the genomic variation that was present at the time, whereas these juvenile trees, which have grown up since ash dieback arrived, they have faced a much stronger selective pressure from ash dieback. And they faced that for two reasons. One is that they are dying more quickly, and the other one is that adult trees, even if they don't die, if they're badly affected by ash dieback, their reproductive rate is diminished. And there's a nice study from Denmark showing this. So they're producing fewer pollen and fewer seeds. And so at these two levels, natural selection should be shifting allele frequencies in the juvenile population so that genes associated with resistance are becoming more common in the juvenile population than in the adult population. So that's what we wanted to look for. So we sequenced over 600 trees. We then looked at the SNP loci that were variable, and we found many fewer SNP loci, single nucleotide polymorphism loci, single letters in the DNA that vary. We found many fewer of them in Marden Park Wood than we had in the mass screening trials. And in many ways, that's not surprising, because the mass screening trials captured diversity from right across the UK, whereas we are just looking at a single wood. So this wood, of course, has lower genetic diversity than the ash population of the whole of the UK. Then for each of our trees, using our genomic prediction models trained on the mass screening trials, we calculated genomically estimated breeding values for each tree. And then we compared those to our phenotypic assessments of ash dieback damage in the wood. Now, we'd kind of hoped, we'd very much hoped, that we would see a really nice correlation between our genomically estimated breeding values and the damage scores, but this is not what we saw. We didn't find that nice correlation. However, what we did find was if we took the extremes of our genomically estimated breeding values, so if we took the third of the trees with the highest genomically estimated breeding values and we compared those with the third of trees with the lowest, then on average, we did see a difference in their health scores. So here for the adults, the low GBV group has got lower canopy cover than the high GBV group, and similarly with the juveniles with their damage scores. And so just as I was saying, even with the mass screening trials, we couldn't get good prediction for individual trees, but we could, looking overall, well, we get some hint from this that these GBVs are actually telling us something meaningful. The models are working in this population. We might think, well, why aren't they working better than they are? Well, if we think about the disease triangle, we've got the host and the pathogen and the environment. Now, in our mass screening trials, a lot had been done to keep the environment similar. So the trials were planted on former agricultural land. The trees were evenly spaced. They're all planted at the same age and at the same time. There's a lot that's been done to make the environment similar, whereas in a natural woodland, the environmental variance is huge. The trees have different spacing, have different shading, they're competing with different plants and different species of tree. There's probably differences going on at the fungal level within the soil. Some of our trees have got canker as well as ash dieback, and incidentally, we're now collaborating with Rob Jackson at Bifor to look at those cankers. And of course, the trees are at different ages as well. So within all this environmental variability, of course, it gets much harder to predict the level of damage simply based on genetics alone, because the environment will be having such a large effect. So then we compared our adults and our juveniles. So we took all the adults and all the juveniles and compared their mean genomically estimated breeding values. And we do see a slight difference. The juveniles have slightly higher GBVs than the adults. Now, this is not a significant difference. P of 0.061. We haven't got to that magic 0.05. But you can see there is a slight difference there. And it would be very nice to do this again with a larger sample size. But that was a very sort of rough and ready way of looking at what's going on. And so what we decided to do after that was say, well, and this figure, it gets quite complicated. What happens if we think of the juveniles not just as a sort of population of their own? Here we're sort of looking at the adults as one population and the juveniles as another population. But of course, the adults and the juveniles are related. A lot of these adults are parents of these juveniles. So what we can say is, well, let's look at how related the juveniles are to the adults. And let's predict the breeding values of the juveniles, not on the genetic loci they actually have, not on their own genotypes, but let's predict them based on which adults they're most closely related to. So we did that, and then we compared that to our genomically estimated breeding values based on the actual DNA of those juvenile trees. And when we did that, we found that on average, our GBVs based on the actual DNA were higher than our GBVs that were predicted based on which adults they were related to. And what that's telling us is that the survivors from the reproduction that's going on of these adult trees are the ones of their offspring that have better genomically estimated breeding values. So the families being produced are being selected so that the survivors are those that on average have the alleles that we think they should have. The alleles of the offspring are not just a random selection of their parents, so natural selection is happening. So we think we've got good evidence for that, but it's a very small effect. And there's a big question in our minds, well, is that effect going to be sufficient for natural selection to do the job for us and breed trees with greater resistance to ash dieback? Especially bearing in mind that as ash trees die off, there's going to be less of the fungus around as well, and so the selective pressure is going to drop off in the wild too. Another thing we did was, for some of the trees, we could identify their actual parents with a degree of certainty. You can never be totally sure, of course, but there were some trees where we could, with a degree of certainty, work out what their parents were from their DNA. And so then we thought, well, let's compare our prediction of the damage of the juvenile trees based firstly on the leaf canopy of their parents, and then secondly on our genomically estimated breeding values of their parents. And let's see which does better. And so in this top chart, you can see the offspring's health in the fields on the vertical axis, and on the horizontal axis, the canopy cover of the parent tree. And there's very little correlation there. But when we use our genomic estimate of the breeding value of the parent tree and the actual health of the juvenile trees, we can see there is a better correlation. So this suggests that our genomically estimated breeding values are actually a better way of selecting trees for breeding programs than just looking at their canopy cover in the field. So there is value in taking the trouble to get a genomically estimated breeding value. So conclusions for ash. We think we've uncovered at least some of the genetic basis for resistance or tolerance to ash dieback. We can predict the health or tolerance of a tree's offspring better using genomics than we can by assessing the leaf canopy of the parent tree. Natural selection is slightly increasing the resistance of ash populations based on this study of one population. And we think that probably a genomically informed breeding program is the best way forward for ash. Now, more briefly, I want to talk about oat, oak and birch. Now, we've been working on oak genomes for a few years now. And one complication, of course, with oak is that we have two common species. We have sessile oak and pedunculate oak. And this complicates genomic studies. And so here's a look at the forest research, forest monitoring plots that they have all over Britain, looking at the oak trees within them. And when they resurveyed these plots a couple of years ago, they sent us leaves from the oak trees. And with funding from Defra, we've sequenced the genome of these. And of course, the first thing we looked at was what species they were. And what is quite interesting about this is, of course, it's showing that Quercus robur is more common in the south and east, and Quercus petrea is more common in the north and west. That was well known. But what we're also able to do with this whole genome data is see hybrids between the two, and not just first and second generation hybrids, but also hybrids that just have had a lot of backcrossing and then just have a tiny amount of one of the parental genomes still within them. And when we do that, we find that there's a lot more introgression of small amounts of Quercus robur genome in Quercus petrea populations than vice versa. So there's lots of Quercus petraea around carrying a bit of Quercus robur, but there's not many Quercus robur around carrying a bit of Quercus petraea. And these hybrids, which are shown in green in this map of the UK, are really quite common and go right up into Scotland. They're not just sort of found around the area where the two species are commonly found together. So that's quite interesting in and of itself. But our main focus is of course a plant health focus, and it's acute oak decline, which Sandra Denman at Forest Research has been working on for some years now. Many of you will know it well, characterized by canopy loss and by these bleeding lesions on the trunk. It's an association of at least 4 bacteria and a native beetle. And we're working on this in association with Forest Research. This is a project now within the Centre for Forest Protection. Action Oak is bringing together lots of different interested parties, and Woodland Heritage have also done a lot of fundraising and partnering work on acute oak decline. And in collaboration with Sandra Denman and Nathan Brown, we've been sequencing genomes of oak trees from sites with acute oak decline. Here's the sites that so far we have data for, or we're in the process of getting data for. And we're aiming to sequence 2,000 oak trees from AOD sites, roughly half of them with acute oak decline and half of them without acute oak decline. And then once we've got that data, we can do a genome-wide association study and see if there are genetic loci associated with having acute oak decline or not. And this will give us a first look at whether there is a heritable basis to susceptibility to acute oak decline. And you might say, well, that sounds a really simple question to try to answer by spending quite large sums of money on sequencing oak genomes. But to actually look at, to use a non-genomic method of looking at the heritability of this disease would actually take many, many decades, because we would have to take clones of oak or families of oak and then grow them up and then expose them to acute oak decline. And to use those traditional methods of looking for heritability would take absolutely ages and be incredibly difficult. So this is actually quite a cheap and quick way of looking for a heritable component of susceptibility to this disease. If we find there's no heritable component, then we know that trying to select seeds or do a breeding program for acute oak decline resistance is not worth the effort, and it's all about the environment. But if we do find loci associated with acute oak decline, that could inform seed choices or even a breeding program. While we're doing this, we realize that, of course, we're mashing up oak leaves to get DNA out, and we don't just get the DNA of the oak, we also get the DNA of anything that's living on or near to the oak leaf. And so Louise Gathercole, a PhD student in the lab, has been using this byproduct of our sequencing to look at the metagenome of oak, the genomes of the other species associated with them. And of course, of particular interest are the 4 bacteria that have been associated with acute oak decline in the past. And so she looked at the presence of these bacteria in our DNA samples, and then she looked to see if they were found on trees that had AOD or that had no AOD symptoms or had had AOD in the past and were now recovered. And she found pretty similar levels of evidence for the occurrence of these bacteria on oak trees of all those different classes. So they're not just found on trees that have symptoms. And then she looked at the different sites, and we have one site that doesn't actually have acute oak decline that we'd sequenced some time ago. This was our first tranche of data this is based on. And in that site that doesn't have acute oak decline, the bacteria are all found. So if this is correct, and if this isn't some sort of effect of contamination in the lab, then this suggests that this disease is caused by widespread bacteria. It's not a case of a novel bacterium spreading and causing a pandemic. There's there's a lot more complexity going on here with this syndrome. So conclusions for oak. Hybridization and backcrossing between Quercus robur and Quercus petraea seem to be frequent, with introgression most common into Quercus petraea. We've started work on the genetic basis for resistance and tolerance to, or tolerance to acute oak decline. And bacteria involved in acute oak decline seem to be widespread in UK oak populations. And then finally, and most briefly, birch. With funding from the Centre for Forest Protection, from Defra, we've started some new work on birch, and we had some large collections in the Millennium Seed Bank at Kew at our Wakehurst site of birch from all over the UK, from natural populations, collected under the UK National Tree Seed Project. And here's Mike Charters, a postdoc funded by the CFP, retrieving some of these seeds from the -20 store underground at the Millennium Seed Bank. And then here are the seeds being planted out by the horticulture team at Kew, so that on a single bench, we've got birches growing from all across the UK. We then collected them and we extracted DNA and sequenced their genomes. And we also did this for alder as well as the various different species of birch found in the UK. So here's a map of all the sites where we've now sequenced progeny from. And we're using the genome sequences from this data to see if we can find associations between the frequency of different genetic variants and the environmental conditions under which these birch trees are growing in, so that we can try to find signatures of local adaptation. And in order to drill more deeply into that, we've also started work on a provenance trial that was set up about 20 years ago by Forest Research on silver birch. And they collected up birch seeds from all of these different sites in the UK, shown by small white dots. And then they planted them out at these 4 sites in trial plantings. And we are now working on the Thetford, Wales and Drummond sites, and we're collecting tissues and extracting DNA. And we're going to compare signatures of local adaptation, looking at natural populations across the UK from our Millennium Seed Bank collections, with the performance of different provenances from across the UK in these different sites with different environmental conditions, to try to really understand the genetic basis of local adaptation in birch. And if we can do this, it means we can inform planting the right tree in the right place in new plantings. And at the moment, of course, we have a fairly crude system where we have these native seed zones, and you need to plant seeds that are from your native seed zones or a neighboring one. But we could potentially get much more precise recommendations about what to plant where, that would hopefully increase the overall health and growth rate and carbon fixation and timber and pulp production of our woodlands going forward. And there's one of the sites that we're collecting DNA from. That's one of the trials. So with birch, a legacy of previous work is giving us a good study system for investigating the genetic basis of local adaptation. So that's the 3 of the systems we're working on. And we're also starting work on elm, but I won't talk about that now. We do have an elm conference at Kew, co-organised with the Institute of Chartered Foresters next week. If you're interested, it will be online, and just look at the Institute for Chartered Foresters website if you're interested in coming to that. But my overall conclusions are, of course, there are many threats to the health of our trees. There's pests and pathogens, there's climate change. We need to be doing what we can to counter these threats, and genomic research may be a great help. It may help us to breed resistant trees. It may give us a better understanding of diseases and their consequences, and help us plant the right tree in the right place to start with. And funding for this work is mainly from Defra, also from NERC. And as I finish, I would just, if I may, put up a little advert for a job opportunity at Kew, Head of Plant Health and Quarantine, a really exciting and interesting role. It's still open, closes on the 1st of July. Someone to run our quarantine house and be in charge of biosecurity for the whole of Kew, making sure that our shipments aren't bringing in diseases and controlling disease within the gardens. So that's a fantastic job opportunity. Please do apply for it. Thank you very much.
Thanks, Richard. I'm so inspired by your talk, which is a humble physiologist. I think I now, and I'm probably being naive, understand some of the plant genomics that you talk about. That was a really excellent presentation. It's inspired me to apply for your job. So,
Please do.
Okay. I do have a greenhouse in my back garden. I don't know if that qualifies me. Okay, everybody, that was wonderful. That was inspiring and informative and entirely relevant to this conference and a great way to bring things to a close. Right, I would ask people for any questions, please put them in the chat or the Q&A. You're obviously so enamored by the talk that nobody's put anything into the talk as we've been going along, but please stick them in now. I'll ask a naive question. All my questions are naive. You talked about the conifer genome being much bigger. Is there any functional significance to that?
Yeah, that's a very good question. We think a lot of that is junk DNA, basically, which of course means that when you sequence, you're sequencing a lot of information that is really hard to interpret. But of course, within that, there will be lots of aspects of it that actually are involved in controlling gene expression and in functional differences. So it just makes them really hard to work on. You know, exactly why they tend to have larger genomes, we're not sure. It's one of those curiosities about plants that there is this huge difference. And there are bold people who are doing genomics of conifers, and I have great respect for them. But I took one look and thought, I don't think I could go there.
I mean, I'm just waiting for a few questions to come in, so I'll hog the questions. But I know you have an interest in the origin of angiosperms and Darwin and so on. Is there anything in the angiosperm success that meant that they were able to get away without that larger genome? Was there anything in that transition?
I think it has been suggested that smaller genomes are part of the key to the success of angiosperms. Because if you can have your genes densely packed into quite a compact genome, you're more efficient, for one thing, because you're not wasting energy, lots of N and P, on very big genomes. And so it would seem that part of the advantage of being an angiosperm is the small genomes. And of course, they have a much more rapid life cycle as well. So it takes a very, if we're looking at pine or spruce, the amount of time they take to provision their seeds and then develop the embryo is much, much longer than your average angiosperm. And so it might even be feeding into that as well.
Yeah.
So I guess one argument is just, well, gymnosperms have just been around so much longer than angiosperms that they've just had longer to accumulate junk in their genomes, whereas angiosperms haven't. But then that also begs the question of, well, why did angiosperms start off with smaller genomes, and where did that come from? So that's an aspect of the abominable mystery itself.
Yes, I've read your musings on that. Really interesting. Okay, we have a couple of questions come in. This one, what kind of coverage are you sequencing to, which is a technical question. And then there's a couple of caveat follow-up questions to this. Also, are you doing some population genetics analysis beyond things like admixture? And how diverse are the two oak species? Sorry for multiple questions. So there's 3 questions there. Are you, what kind of coverage are you sequencing to? Are you doing some pop gen analysis? And how diverged are the 2 oak species?
Yeah, so our sequence coverage varies in direct proportion to our funding. In the oaks, we're doing 20x coverage, which is pretty good, and should enable us to have pretty accurate SNP calls at the variable loci. In Marden Park, it was more like 10 or 11x coverage, which is less ideal, and that has introduced a bit of uncertainty into some of our allele calls, which we've sort of regretted since we did it. In our initial GWAS on the mass screening trials for ASH, we realized that if we sequenced individuals individually, we would get really low coverage with the funding we had. So what we did was we made pools of all of the healthy trees from one provenance and all of the unhealthy trees from one provenance. So 2 pools per provenance. And then we sequenced the pools. And so that gave us estimates of the allele frequencies within those groups. And for a GWAS, that is all you need. And so it was a really cost-effective way of doing quite a powerful GWAS. And that's something we've not had any regrets about doing. That works really well. So there's different ways of designing things to fit whatever budget you happen to have at the time. For oak, yes, we will be doing more population genetics. So Louise Gathercole has only just started analysing that data set from the Forest Research Oak Condition Survey, and there'll be much more coming through. Of course, the first thing we had to do was work out what species was what, but certainly there'll be a lot more coming through. And then the third question.
How diverged are the 2 oaks? I mean, they're coming together as hybrids, so presumably not totally diverged.
No, they're not totally diverged, but in the oak phylogeny, interestingly, they're not sister species. They all have closer relatives. They both have closer relatives. In both cases, species that are found in Southern Europe. But they are fairly close relatives, although they are not the closest relatives.
Okay, and then a question from Charles. Will the host evolve faster than the pathogen? So is there an arms race going on here that if you waited for some time, you'd see the defenses then be overcome again?
Yeah, well, this is a real conundrum with trees, because you would think, well, the pathogens have generation times that are so much shorter than the generation time of the trees themselves. So how do we even have trees, given that presumably they must evolve so much slower than the pathogens? One thought I have on that question is, well, maybe the tree's strategy is that they sort of soldier on for long enough that the pathogen itself starts to attenuate its pathogenicity before it's finished off the tree population. And so the trees just try and sort of hang in there for long enough for they're not evolving, but the pathogen is, and the pathogen is actually getting less harmful. Maybe that's how they do it. I think another thing is they just harbor so much diversity within their populations. And it takes such a long time to finish off a population that perhaps you can see some fairly major allele shifts that happen. But then when the epidemic subsides, you've still got pockets of that residual genetic diversity that was there before that can then feed back into the population. But it's a real conundrum, the whole idea of an arms race between an oak tree and a bacterium or a
Yeah, fascinating. I hadn't thought of it like that before, so it's an interesting question. So when you were talking about the juveniles versus the mature trees, you sort of said it was the spores getting into the canopy. Is that penetration into the leaves in the canopy? Is that penetration of the spores, is that the key sort of physiological battlefield of defense, or is there something subsequent to actual pathogen invasion?
I think that's just one of them. And then the other major one is the transition from the leaf into the woody tissue. And I think that might be the more important one. And I think some trees probably cope quite well with ash dieback by losing their leaves before the fungus manages to get to the woody tissue. And very interestingly, a small number of the adult trees with our best genomically estimated breeding values had really bad canopy scores. And we wonder if what might be going on is that those trees actually are really good at resisting ash dieback because they're losing their canopy. I mean, it's a bit of a sort of a posteriori hypothesis that's trying to explain away a slightly tricky aspect of our results, but it is quite a tempting hypothesis that maybe that's one of the strategies that a tree can take.
So that's an advantage of being deciduous.
Yeah, definitely. Yeah, definitely.
Yeah. So when you talked to, I haven't got any other questions coming in. Maybe we should be thinking about finishing in a minute. But when you talked about you had the genes and gene annotations, and you were picking up things like salicylic acid and so on. Are there any better clues in the annotation, or is the genome annotation not precise enough to give you those sort of clues that you'd be after?
Well, you just have to take them all with a big pinch of salt, because often this will be this is what the gene's doing in Arabidopsis, but how do we know it's doing the same thing in an ash tree? So we do have to be really quite careful around that, and a lot of the genes we just have no idea what they're doing.